My name is Seyed Ali Elahi and I'm a postdoctoral researcher at KU Leuven. The title of this presentation is In Silico Techniques as a Bridge between in vitro and in vivo insights in cartilage mechanobiology research. The use of in silico techniques as a framework to integrate insights from in vivo and in vitro research is central in our ongoing Happy Joints project. This is a multidisciplinary research line headed by Ilse Jonkers, and it runs between the Human Movement Biomechanics group, the Biomechanics division, and the Tissue Homeostasis and Disease Research group at KU Leuven. Within this presentation, I want to highlight our multifaceted research approach and the role of in silico approach to bridge between experimental insights. I have no conflict of interest. In the Happy Joints project, the aim is to optimize joint health throughout lifespan. Therefore, we aim to elucidate the factors that will contribute to joint happiness. In this, we take a mechanobiological approach, aiming to further insights on the role of mechanical stimuli in cartilage biology. From these insights, we then aim to further the understanding of the mechanobiological factors contributing to cartilage disease and in particular, early osteoarthritis. Indeed, during daily locomotion, the joints undergo dynamic mechanical loading. As an example, here we can see the movement of knee human joint. The cartilage layers in the healthy joint consist of multiple cartilage layers that act as a contact interface between the ends of the long bones and provide nearly frictionless movement. The role of cartilage is to allow a smooth movement of the contact surfaces and to distribute the loading over the contact surface. As such, it is crucial in absorbing shocks, as for instance at heel strike during gait, and in minimizing peak loads to the underlying subchondral bone. In a healthy joint, cartilage is in homeostasis and the mechanoresponsiveness is optimally tuned. This means that the cartilage structure is optimized to its mechanical environment and the biology is optimally tuned to maintain this structure. A joint injury will alter the mechanics of the joint drastically. Rupture of a ligament or a cartilage injury will alter the local mechanical environment of the cartilage and will upset its biological response, thereby causing progressive degradation of the cartilage and triggering undesired protective responses of the subchondral bone that further alter the mechanical environment of the cartilage. Furthermore, genetic predisposition and epigenetic changes can affect the cartilage biology and cause deterioration of the mechanoresponsiveness. Often both mechanical and genetic factors coincide and underlie the onset of degenerative joint disease. In the Happy Joints project, we focus on understanding the role of mechanical loading in articular cartilage homeostasis and degenerative joint disease. From this, we aim to provide insights to optimize cartilage regenerative treatments such as physiotherapy and tissue engineering approaches. To elucidate the role of mechanical stimuli in joint health and disease, we need to complete the knowledge circle that starts from investigating the moving joint and ends with relating it to the biological responses of the chondrocyte cells. This requires a multifaceted approach that combines mechanical and biological insights that each are derived using their dedicated experimental approaches, and where in silico techniques will allow to bridge across multiple scales. I will use this scheme originally proposed by Andriacci to structure our own research work on cartilage mechanobiology. In order to understand the moving joint, we will perform in vivo motion analysis to measure the joint movement and ground reaction forces during different activities of daily living in healthy control subjects and subjects with degenerative joint disease. Then we can use in silico musculoskeletal simulations to determine the internal joint forces during locomotion and evaluate the biomechanical fingerprint of locomotion. Next, we can use structural analysis techniques to evaluate the local deformation and stresses of cartilage tissue surrounding the cells that represent the relevant microenvironment. Using in vitro experiments that involve multi-axial bioreactor experiments, we can then evaluate the effect of different aspects of the macro and micro environment on the biological responses of the cells in the cartilage. Only if this circle is completed, we can achieve our ultimate aim, to predict the effect of traumatic events or therapeutic interventions that alter joint movement on disease onset and progression, or alternatively evaluate the success of a cell-seeded scaffold to arrest local cartilage degeneration and initiate repair. I will now present the research in the Happy Joints project in each of the three subdomains. The first are the in vitro experiments in progress in THD Research Center, where the focus is on documenting the biological response of cell-seeded hydrogels and cartilage explants when loaded within multi-axial bioreactors under different biological states. The second subdomain relates to the in vivo experiments in HMB group, where joint movement and cartilage constitution in humans, but also in animal models using motion analysis and imaging methods. In order to integrate the insights of both domains, in silico simulations will be performed as a bridge between in vitro and in vivo experiments. To this end, we use in silico techniques to characterize the local mechanical environment during the in vitro bioreactor loading experiments and relate these to the observed changes in the biological parameters. As such, we define mechanoregulatory rules representative for the effect of the microenvironment on the cartilage constitution and structure. Furthermore, we will use in silico techniques in combination with the in vivo data to determine the joint forces and local cartilage loading during movement in human and rodent models. First, I briefly present our in vitro experiments and results. In our in vitro experiments, we load in a controlled manner cell-seeded constructs and explants. For this, we retrieve cartilage plugs from the knee joint and harvest the chondrocytes. These cells are responsible for producing extracellular matrix, which mainly consists of collagen fibers and proteoglycans. Changes in the mechanical environment of the cells, for instance, due to a defect in cartilage, change in the loading condition, or change in the biological state of cells can impair the constitution production, lead to progressive degradation of ECM and cell death. If we fully understand the responses of cells and ECM to mechanical loading, we can intervene and alter the mechanical environment in order to optimize the regeneration of ECM. In our current work, we isolate cells from cartilage biopsies in patients undergoing total knee replacement surgery and seed them in 2.2% alginate hydrogel discs. Then we put the cell-seeded constructs within our torsion compression bioreactor setup to simulate different multi-axial loading representative for the multi-axial loading conditions in the knee cartilage during daily activities. We then evaluate how ECM production is affected by the different loading protocols. First, we focused on uniaxial loading conditions in which we compared 4 different loading magnitudes, dynamic compression of 5%, 10%, and 20% at 1 Hz for 1 hour and a control sample. We then evaluate the cell response in terms of the gene expression related to the production of the major ECM constituents, but also matrix degrading enzymes, indicative of matrix breakdown. For instance, the results show that the compressive loading magnitude did not importantly affect COL2A1, although with 10% compression being the most effective. For ACON and PERLECON, 20% compression was found to be the most effective uniaxial loading condition. Once these relations were clear, we investigated if the presence of shear strain would alter the response to loading. Therefore, we applied 10 degrees torsional loading in isolation as well as 10% compression combined with 10 degrees torsion. Interestingly, the previously observed downregulation of COL2A1 was not altered when adding shear loading. In contrast, the previously observed upregulation of ACON and PERLECON under pure compressive loading was lost when adding shear. Likewise, we have now documented the effect of additional shear load on matrix degrading genes and growth factors in cell-seeded constructs and are now considering loading explants. Our current research insights document the mechanobiology of healthy chondrocytes and allow to determine the critical mechanical stimuli associated with the switch between anabolic and catabolic molecular responses of cartilage tissue. In ongoing research, we are now manipulating the cell phenotype to represent signatures of early OA to extend our insights towards mechanobiology of OA cartilage. Our current insights indicate that excessive WNT signaling induced by CHIR stimulation alleviates the mechanoresponsiveness of chondrocytes under compressive loading merely through impaired SMAD2/3 pathway activation. Although these analyses provide us insight in how altering the mechanical loading affects the cell responses, they do not relate them to the local mechanical environment of the cells in the hydrogel or explant. In order to understand this, we need to relate the applied mechanical loading to the local deformation or strains and local loading or stresses in the hydrogel and explants using in silico simulation. The finite element simulation of bioreactor tests requires specific inputs related to geometry of samples, which in our case are cylindrical, the applied loading, i.e. uni or multi-axial, and the mechanical boundary conditions being unconfined compression. Furthermore, we need a material model that describes the mechanical behavior of the cartilage and hydrogel that we define it in a collaboration with University of Eastern Finland. The material model requires accurate experimental measures of material properties. We used dedicated modeling techniques combined with a sensitivity analysis to determine the optimal experimental testing protocol for the mechanical characterization of cartilage and hydrogel constructs. We found that a single-step relaxation loading of 20% compression with 10% per second rate was most sensitive to describe the mechanical behavior of the hydrogels, whereas a two-step relaxation loading with 10% compression in each step and 10% per second and 1% per second rates between the first and second steps was most sensitive for cartilage explants. These experiments will now be used for our further experimental work. Finally, having the finite element simulation results. Here you see the displacement fields under compressive and multi-axial loading, indicating maximum deformation on the edges of the top surface of sample in the multi-axial loading in comparison with the uniform maximum deformation on the top surface in the uniaxial compression. Using these mechanical deformation maps, we can now begin to better understand the localized biological responses observed in the bioreactor experiments. Furthermore, we can use these models to generate insights to determine the loading protocols and the hydrogel material properties in order to optimize regeneration and limit the degeneration in cell-seeded hydrogels. More specific, using the in silico approach, We studied the effect of different compression and torsion loading protocols as well as the stiffness and permeability of the hydrogels. We varied these parameters in a predefined range and evaluated their effect on ECM regeneration and degeneration in terms of maximum principal strain and fluid velocity. These analyses show that by increasing both compression and torsion loading in the defined range, both ECM regeneration and degeneration increase, whereas increasing the stiffness and permeability of hydrogel would increase the ECM degeneration. From these results, we concluded that within the studied range, using the maximum torsional and compressive loading combined with low stiffness and permeability would be preferable. We are currently integrating these insights in the development of new in vitro protocols as well as in the development of next generation hydrogels to support chondrogenesis. The challenge now is to evaluate if these mechanoresponsive rules are also underlying degenerative joint disease in vivo, knowing that these conditions are multifactorial. We aim to elucidate the mechanical environment of the human joint during locomotion. Taking the step from the simple geometry and loading conditions in the bioreactor to the human joint with a complex geometry and constitution and highly complex loading conditions is not trivial. It requires combining in vivo motion analysis and in silico simulation to elucidate joint loading and ultimately cartilage deformation during locomotion. For these in silico simulations, we require information on joint geometry that can be readily obtained from state-of-the-art in vivo imaging methods. MRI is typically used to obtain subject-specific information on 3D cartilage geometry and thickness. In addition to geometry, using in vivo imaging like dedicated relaxometry sequences, information on cartilage constitution can be derived that can then be related to more complex cartilage models that account for the content and distribution of the different constituents to describe the local material properties. Typically T2 and T1 row sequences are used. The in vivo loading conditions during locomotion are calculated by combining musculoskeletal models that describe the geometry of the lower limb musculoskeletal system with 3D motion capture data. To perform the 3D motion analysis, we use reflective markers on the volunteer's body to record the 3D position of the body in space. We also use embedded force plates to measure the ground reaction force during movement. Here you can see how the movement of the subject is measured together with the interaction forces between the subject's foot and the ground. This information documents the subject-specific movement characteristics and potential movement compensations. The subject-specific motion analysis results are combined with a MSK model to calculate the internal joint force during movement. A MSK model describes the geometry of musculoskeletal system, the muscles, bones, and joints defined using imaging methods. When analyzing knee joint loading, we incorporate a complex model that accounts for the multiple degrees of freedoms, the different ligaments, as well as the cartilage to cartilage contact. This model is first scaled to the size of and mass of our participants using the motion capture data. Then we calculate the body motion from motion analysis results. In combination with the information from the ground reaction forces, we can then obtain the forces of the individual muscles, but also ligaments required to produce this movement. This will finally provide us with information on the magnitude of the contact pressure in the joint as well as its location. The obtained pressure is then applied as boundary conditions in the finite element modeling of the knee joint to obtain the local cartilage deformation and local loading. The information on this local mechanical environment can be related back to the cartilage composition observed in the MRI and ultimately the in vitro experiments aiming to establish mechanoresponsive rules underlying cartilage constitution in vivo. We found that in healthy knees, areas with higher compressive loading during gait were related with thicker cartilage with higher proteoglycan and collagen content. In contrast, areas subjected to higher shear forces presented lower proteoglycan content. These results are in line with the observations of our in vitro bioreactor results, which is encouraging. Using the complex knee model in combination with in vivo 3D motion capture, our group showed that already in patients with early OA who only present signs on MRI but not on radiographs, the loading magnitude in the medial compartment was increased during gait. Furthermore, we found that the location of the loading was shifted away from the physiological weight-bearing area in the center of the medial plateau, more towards the posterior and lateral aspect of the cartilage surface. This confirms that even in the early phases of the disease process where no radiographic changes are present, the mechanical environment is altered, confirming mechanical loading to be an important contributor to early progression of the disease. In our most recent work, we are extending our in silico work towards animal experiments. In the context of degenerative joint disease, rodent models take a prominent place and are well established. Compared to in vivo human research, animal models are highly controlled and allow detailed documentation of the cellular and tissue characteristics throughout disease progression. This allows to further elucidate the role of mechanical and biological drivers of cartilage homeostasis and disease. Therefore, in recent work, we are translating our modeling workflow for use in rats to document the mechanical joint environment in the presence of mechanical knee instability to elucidate its role in the onset and progression of cartilage degeneration. We are acquiring high accuracy data on the rat locomotion using a dedicated biplanar X-ray setup to measure 3D knee kinematics and a force plate to document the interaction forces. This unique setup is available at the University of Antwerp. Here you see the unique acquisition of skeletal movement and ground reaction forces. Currently, we are working on customizing our in vivo workflow to combine the 3D motion capture data with a dedicated rat model to calculate the muscle forces during locomotion. Based on these loading conditions in the knee and ultimately the cartilage strains will be calculated and related to the disease progression as observed in terms of histological and molecular changes. With this overview, we presented a selection of our past and recent work on how we relate our in vitro and in vivo experiments using in silico simulations to further our understanding on the role of the mechanical factors in cartilage homeostasis and disease. So far, the modeling approach has been a solid bridge to integrate scale-specific experimental insights, allowing us in the future to bridge our understanding on mechanobiology from cell to the joint. With this, I would like to conclude my presentation. We are grateful of our funding sources, KU Leuven internal funding, FW funding, and our Marie Curie European Fund. I also would like to thank our collaborators at University of Eastern Finland, University of Liège, KU Leuven, UC Leuven, Stanford University, University of Colorado Boulder, and Antwerp University. And finally, I want to acknowledge the work of our group members, PhD students, postdocs, and PIs. If any question raised for you during my presentation, please contact me by the following email. Thank you very much and hope to see you next year in Rome.